Hello, this is Mr. Whaley for the Science Department at Manor Senior High School, and I'm going to talk to you about your Biology IGC project, The Checklist. This is the secret to being successful. It's no secret though. It's a breakdown of the project. If you do this, then you will graduate. You can see 4-week IGC plan, week 1, April 5th to 9th. Teacher and students introduce IGC explain checklist and assign checklist. So these are the parts of the checklist you need to finish by the 9th of April. Now go ahead and work on it in the weekend, right? The 10th and 11th, but make sure you get that much done. And then April 12th through 16th, you should finish these parts of the checklist. And then week 3, April 19th to 23rd, that checklist is all done and you're working on your presentation. You have one of these. This is mine, and I'm going to show you an example here. So name the virus. You notice I don't have any sources linked here. I haven't taken the time to do that because I'm confident you can, and with your teacher you can work on that. But I've written down as if, imagine I imagined the SARS-CoV-2 that caused the COVID-19 epidemic or pandemic all over the world. SARS is because sudden acute respiratory syndrome. Sudden means surprise, acute means sharp, respiratory means origin of infections in the respiratory system. Syndrome means a lot of symptoms showing up together like loss of taste, shortness of breath, and fever. And the CoV part of the name, SARS-CoV-2, is because its shape is a bunch of spikes making like a crown around the round virus. And then CoV-2, because this is a new coronavirus to cause a pandemic. The first one was back in 2002. It was just called SARS-CoV. And so that's something of what this will look like. It'll be based on the shapes of the thing. That's the CoV part. It might be based on symptoms that people have. So imagine like Ebola 2, right? But what does Ebola mean? So find out what the relationship is. Your job isn't to just say virus part 2, but it's going to be related to some virus that's already out there. Okay, explain the difference between the structure of a normal cell and this virus. You can see my link there, one of my sources that I'm going to use to explain this difference, and I'll just show you where that link goes. It's in my playlist that I shared with you. And it's this Amoeba Sisters video, and you'll notice video intro, and the second part, it says down there real small, it says intro to a virus. And then actually some of the other things you might need, virus structure to explain this, the differences. And then the next one, the next 2 parts are about the replication cycle. So I want to show you where this playlist is. Scroll down to the bottom of your checklist. Underneath that table, you'll see helpful links, and I put video playlist right at the top, right here, because other teachers might find something that's helpful and can tell me about it, and I'll add it to this playlist. If I'll go back to the actual literal playlist.
Once there are many copies of the viral DNA,
oh, that's another video in the playlist.
Reassemble and form new viral particles.
And so here you can see those videos are found in here. Here's the Amoeba Sisters one. And here's the naming one. So I would take this one and I would put that in my, I would go to this and copy and paste that link and put it in my cells up here by naming for my resources up here where it says name the virus. I'd put my link right there for that other video. And I would start putting this project checklist together, start taking notes on this information. I want to talk about this one for a second. This is the one you probably need the most help with. Describe how the virus reproduces, or another word is replication. So here's a YouTube video from my playlist. It's also down below here. And then here's an animation. That's what this picture right here is. And I wrote this down from this video. Viruses don't have ribosomes, so they need the ribosomes of living cells to translate their instructions into new copies of viral proteins and shapes. So that's the why of the process, but I want to tell you the how. I'm describing it. If you want to describe a process, you can't do it unless you use verbs. And you got to use a lot of verbs. So if somebody asked me, what did you do today? If it was a process, I'm not just going to say, I just did stuff. That doesn't describe a process. And so I need verbs. And so in this case, this is how the virus does its job. It attaches, it penetrates, it uncoats itself, or it gets uncoated, and then it is replicated, assembled, and released. So every virus goes through this process, and then some of it might be a little different, like the attach part might happen a little differently with each one. So in this case, the virus has a protein, well, in general, the virus has a protein on its outside that is shaped in such a way that it attaches or binds to a protein in some of the cells in our body. So I'll give you my specific example of mine, SARS-CoV-2, if that were the one I invented, which I did not invent it. Then down here on this question, how does the virus cause disease, the mechanism, that's where I would put the same as this attach, but I want to put how it attaches. What's its special mechanism? And so for the SARS-CoV-2, the special mechanism, it is a spike that binds to an ACE2 receptor on human cells. That's the name of it. It's an ACE2 protein, and it has an affinity or an attraction so that it's kind of like Velcro. If it touches it, it just binds up to it. And then in the penetration time, it gets fused. So you'll see here the envelope gets fused. Well, how it gets fused is the spike protein gets changed when it has that interaction. So that's what I'm going to put down on my specific mechanism for my virus. I'm going to type more details about how it's special compared to a lot of other viruses. It does this special particular thing. And so on. Uncoat, there's some kind of an opening up. Like I said in the one that I studied, it got fused together, and so all of its mRNA or messenger RNA, so it has the code, the message, and it dumps it in to our cells. And then now the cell starts doing work. That's why I made this a different color, these steps. The human cell's ribosomes translate the instructions into amino acid chains that make up the proteins of the virus. And so it just rebuilds all the parts until then our bodies put those parts together. They just keep following instructions until it's assembled into the new virus. The human cells have other organelles that in them that put those proteins together into the new viruses. They just follow instructions. And then there's a release that happens. The cell releases it. And you'll learn from the different videos, there's different ways that might happen. It probably will just destroy the cell. I mean, imagine if you just made 1 million copies, even if they were really small, it would use up all the energy of that cell. So it's kind of like its life is shorter, and none of its work was to help keep you alive. All of its work was to multiply, replicate the virus. And so at any rate, this release that happens, it's painful, or it hurts our body, I should say. It's harmful. And that's the end of that. Describe how the virus reproduces. That's in general how that works. So here's another diagram I got. So I know that it's useful. Us teachers know, like, look at a bunch of different pictures for understanding this process. So I shared a video with you, and I've showed you 2 different pictures. This diagram has labels on it: attachment, penetration, uncoating, gene expression and replication, and then assembly, and then release. So sometimes the words might be slightly different. I can't remember which word was different. Between uncoating and assembly, this says replicated, and this you have to kind of look to find the word replication. It has other words as well. And then this one doesn't even have words on it, but I can find each of those things on this one. This is specific to SARS-CoV-2, but I can kind of look through and see how each of these things happen. Now this looks a little bit different to some of the pictures I've shown, but we're not looking for perfection. We're looking for you to show your teacher that you can describe something, and in particular, you can describe a process using verbs. You can find the verbs. You can recognize verbs by, oh, this is the same meaning, like attach and bind. That kind of goes together. You're showing us that, and we're able to translate that into, you get credit for biology. You get to graduate. So best of discipline to you and best of organization so that you're able to fulfill your requirements. And hey, be in touch with your teacher. They are more than happy to help you, eager to help you to get this done and be successful. Thank you very much.